Hi, this is Dr. Ben Morrill. Welcome to Reptile Genetics Weekly, episode 27. Last week's didn't count because it was short, and we were trying to catch our breath from everything. Thanks for your patience with us. Sorry we were late last week, and as far as I know, we're planning to be back on Tuesdays next week. I think this is 2 or 3 weeks in a row where we haven't been on Tuesday, but we are planning to go back to Tuesdays. So for today, we're doing a live. I'm here live with you, and you'll be able to cheat ahead a little bit. I've got some notes on the board up here. Oh, it's hard to point backwards. There we go. So you'll be able to see some notes there that I took, and zoom in there, make sure, hold us accountable that we get to everything. So first of all, for results for everybody, so you know when results are coming. We have another big batch. First of all, thank you. We keep thinking that things will slow down, but this is another huge run. Big enough we're actually going to break this big run into 2 runs. I'm going to start a run and then 2 days later start another run. So we will have the next batch of results start going out on Tuesday. So this is Wednesday when we're recording this. So next Tuesday, the first results will start going out, and I'll probably have some going out pretty much every day through the weekend to be able to get through all of this. We have the most panels we've ever had, and submitted. There's lots of people wanting to get full panels run, and we're very thankful for that. And we'll talk a little bit more about that later, how it's starting to get kind of complicated now that we're over 20 tests. But anyway, thank you so much for using the testing, and we are definitely learning, growing, getting faster and better. And today we'll talk about some specific things that we're looking forward to adding and ways that we'll get faster. We talked a couple episodes back and gave you a question of whether you'd rather us be faster or develop more tests, and I was kind of surprised that it leaned more towards more tests than it did faster. So that's to me, that's really cool, because selfishly, that's what I want, is lots of tests. So, so anyway, we've got more testing coming, more test results coming next week. Like I said, through most of the week next week, we'll have results going out. And yeah, so we'll bring Kayla and Sean on and start hitting some of these other topics.
What's up, guys?
What's up? Live from my basement.
Live from Sean's basement.
That's right.
I
we've
told
got
him it was
lots of
actually
people.
interesting to look at. Like, I liked it better than the background you had before. I was like, look at all the neat stuff in there. You can
I actually have a very nice house. Just this is my basement. That's why I usually have the background with all the dragon heads, but it just wasn't working very well today. So you guys are stuck with all my nerdy man cave backgroundy stuff.
We appreciate a nerdy man cave. So.
And thanks to all of you that got on here at 7 or even before 7. I see Blackjack Reptiles, High-Vis Pythons, H&H Pythons, Sloan Morphs, Dark Cloud, several of you, H&H, H&B, HB Reptiles. So yeah, thank you for all joining in. And before, when we've done lives, I've been able to do it on Instagram as well. This time I won't be able to, because I'm actually using my phone to do this time. Usually I've been on my computer and used my phone to live stream for Instagram, but maybe we'll figure out how to add this later on Instagram for those of you that prefer watching there. But anyway, yeah, we've got you Facebook and YouTube. Yeah. Cool.
So if you're following either one of those, you'll get the notification from it. So yes. Awesome. So what's our first topic there on the whiteboard we got back there?
All right. So we have up there many thanks for those of you that came and said hi at the shows. So we had back the furthest, we had Pomona, then we had Daytona, and then Arlington and Tinley. So we got to meet lots of you that have ordered tests. We got to meet you face to face. Some of you I've known for years and sold animals to and bought animals from, and many of you is the first time meeting you. And I think same with Kayla and Sean. It was really fun meeting everybody and talking.
Yeah, definitely.
Yeah, I just wanted to say about Tinley, I had a really good time. That was our sort of 1-year anniversary breaking out into ball pythons. We've been doing colubrid sexing for a few years, but this was really the big push to try and get Dr. Ben to leave his day job and come do this full time. And it worked, right? So we're doing it full time now. And being the customer service guy, I'm always the one dealing with issues and things like that. So sometimes you're feeling like, man, the world's coming to an end today. Things just aren't working out. But then you go to Tinley and you talk to everybody else and you're reminded that, no, what you're doing is really useful and helpful. And it was just really gratifying and rewarding. And so everybody that came up to us at Tinley, particularly me, I needed the boost. Thank you very much. It was good to hear from you all and hear about how supportive you guys have been.
Yes.
Have a little.
Yeah, it was very uplifting. That's for sure. Lots of support and excitement, and just got a message from Antoine saying, "Hope we like the charity event." That was definitely awesome. Seeing the grappling event, and they collected some more money to help USARK, I believe. So
awesome.
That's cool. Yeah. All right. So you think we're close enough with Orange Dream and Leopard? We can give people an estimation of when those will be up, Sean?
Yeah. So we're pretty confident. All of our tests have come back pretty much 100%. So we're going to ask the vendors to release those next week. It'll be up to them how quickly they do that. I know MorphMarket will probably jump on it pretty quick. I think Clutch may be a week or two behind, just because I know they're just about to finish up like a complete software rebuild, which we're going to bring up a few times throughout today's live cast. So that'll probably slow things down a little bit, but I don't hold that against them, because what they're building is really cool. And if we got to delay a couple weeks there, that's no problem. So, but yes, Orange Dream and Leopard are about to pop. So I actually have some people that have known these were going to come out for a while. So we have some sheds that have already started coming in. So if you guys have them and you want them tested, send them in. We should be good to go. Ben, you might want to talk a little bit about the Super Orange Dream versus Orange Dream, and because I'm going to get those questions, and I know you're going to get those questions, and it'd probably be a good idea to just answer it out loud right now.
Yeah. So for me, myself, the animal we have on the screen, I was interested. He was possibly, I was pretty confident he was at least Orange Dream and could be Super Orange Dream. And then I knew he was Het Clown and Het Pied, but he was also a Posset G-Stripe. And so we did run the test on this guy, and he is a Super OD, and he's a leopard. I already knew he couldn't be a Super Leopard, but and then he also did test positive for Het G-Stripe. So he's a triple Het, and he's got Orange Dream, Pastel, Fire, Leopard. I might be forgetting one, but anyway, kind of a fun
Pied,
triple
I think
Het
you
boy.
mentioned.
What's
like
that?
Pied?
Yeah, he's Het
Pied?
Pied. Yeah, Het Pied, Het Clown, and Het G-Stripe. So
yeah.
And then as on the leopard side, it definitely, our leopard test will detect if you have a super leopard versus a straight leopard. So same as any of the other tests that we have. If there's one copy there, we'll be able to see just one copy. If there's two, we'll be able to see two copies. If it's a incomplete dominant, we'll say we'll still call it a het if there's one copy. And that's kind of how we talk about it with genetics lingo. So if it's one copy of Orange Dream, it's going to say heterozygous, and if it has two, it'll say homozygous, which is a super OD. And the same with leopard. If it's heterozygous for leopard, that's just a normal leopard. And if it says homozygous for leopard, then you have a super leopard.
Okay, so the next question, also related to OD, is again something we should probably talk about here, just so that we all are on the same page. What about high OD?
So high OD, I am actually getting a whole bunch of sheds from a few people coming in soon. I haven't tested that in depth yet. From what I've seen with people's breeding and in talking to people, my guess is there's a second gene that's close to Orange Dream that really makes it pop like that. I think it's a disconnected thing that is close enough that it travels together a fair amount of the time, but not all the time. But we'll have to figure that out. So my guess is the high ODs, as far as the OD part, it'll test the same. That's my hypothesis right now. We have to test that. So I don't know that for sure yet. But that's what we're thinking. And then if we get enough high OD sheds in, hopefully we can map that second, like what I'm saying, hypothesis, my guess, if there is a second gene close by that really makes OD pop, then we'll be able to find that as well. And then down the road, hopefully we'll have a high OD test also. But for right now, it'll just be Orange Dream versus non versus, if it's a super OD.
It looks like High Viz Grays joined us. Hey guys, what's up? Thanks for coming. High Desert's here too. What's up, Antoine? That's actually a pretty neat question. Have we seen a lot of super leopards, or is it pretty uncommon?
So I've tested 2, and they came out, or I tested 2 that came out as super leopard, and I contacted the person that those animals came from, which he may do a video specifically about that at some point, but they're very cool animals. But anyway, he did say, yeah, they were possibly super leopards, and one of them happened to be gravid. Well, not gravid anymore. They're eggs that were about to hatch, and so like 3 days later, the eggs hatched, and all the babies had leopard in them. So that also verified that super leopard test was correct, and that female is a super leopard. So there have been some, but not a ton.
Mm-hmm.
Okay, what's next?
Leopard footage. Yeah.
All right. So after Orange Dream and Leopard, we have the Blue-Eyed Lucy complex coming through that Dr. Seidel and the Eastern Michigan group figured out. And so we will have those tests validated here pretty quick. And within a week or so after they're validated, they should be added to MorphMarket and to Clutch. So those are coming soon. And then I know people in the comments have been asking about Puzzle, especially. Also Monarch and TSK Xanthic, all 3 of those. We do have the data. It's being crunched. And so I'm at the point where I'm going to, within the next few days, I will be able to design the test and test them out. And if I can get them on the first try, then those will be very quickly after the Blue-Eyed Lucy complex that I'll be able to validate them as well. And like we talked about an episode or 2 ago, when I say to validate, that's where I want to run 100, where I know what the answer is. I know what the animals are, and I just make sure the test works properly, and we have 99% and above accuracy. So Puzzle, Monarch, and TSK Xanthic will be shortly after the Blue-Eyed Lucy complex. So we got OD Leopard, then the Blue-Eyed Lucy complex, and then we'll have Monarch, Puzzle, TSK, Xanthic. And I don't know for sure which of those 3 will be first. We'll just depend on how lucky we are with them. Hopefully we can get all 3 on the first try, but that doesn't always happen.
I'll be happy with 2 out of 3 on the first swing.
Yes. And then the others that we had listed, we hope to have by the end of October, like GHI and Fire and Pastel. There's still some more of those that I am very far. I mean, they've looked good. Like I've run 15 or 20 or something like that. I just haven't been able to finalize those yet because we have so many other things going on. But the Blue-Eyed Lucy complex is pretty important because there's 5 or 6 genes there, so that'll be awesome. And then the Puzzle and Monarch and TSK we've been talking about forever and waiting for forever. So I want to push those through. But then I'll get back onto some of those others we had listed to try to have done by the end of October.
All right. See, something about Ultramel?
Yes. And so with Ultramel, I wanted to announce, I think we have talked about it before, but we still get questions. We do test for both Ultramel A and B, so the 2 different mutations. We can test for both now. So for quite a while, we could only test for one, the one that Dr. Seidel published. But now she's figured out a way to test for the second one. So any Ultramel or panels that include the Ultramel test, we will be testing for both. So if you get a result that says het or says negative now, that's a negative after testing both. And if it's a het, then it could be het for one or the other. And so yeah, we've got that going in Ultramel. I want to make sure everybody knows because that took a while to get, but we're there. Oh, I lost you, Kayla.
I hear you, Kayla.
Oh, that's because I muted myself while I was typing. So you didn't hear my crazy mechanical keyboard going off. High-Vis Pythons wanted to know if have you noticed any phenotypical differences in Ultramel A and B?
I haven't myself, but I don't see the animals. So if that's something people are interested in, you could certainly reach out and say, hey, are mine one or the other or both? In the United States, by far and away, the more common one is Ultramel A. It's fairly rare that I see B. I would say 1 out of 10, maybe 2 out of 10.
Yeah, it's about that. It's very rare.
Wow.
Yeah,
Okay.
so I definitely do see it. We do need to test for it, but it's much lower frequency than ultramel A. So
I would like to see what a homozygous B looks like.
Oh yeah.
Just because.
Since it's a mutation in the same gene, they both make the gene non-functional, I believe. So I would imagine they'll act the same, but all of the yellow belly mutations are in the same gene and they do different things. So it's possible if someone had a homozygous ultramel A and a homozygous ultramel B, that would be cool to see. So I'll keep an eye out for that.
Yeah, definitely. All right. So
yeah, another thing that we're working on, besides the ball python morphs, as many of you know that have watched the show, we're very interested in other species as well. And actually, the one Sean was just talking about, Tinley, there's a year anniversary from when we started kind of opening up our doors and doing morph tests, stuff like that. It's actually also another anniversary from 2011. It was October Tinley where we started talking about any DNA testing, period, Sean and I, and it was because of green tree pythons, and we wanted to make a sex determination test. And so in this last batch of data where we got the TSK, Xanthic, and Monarch and Puzzle and all these other things we've been waiting for, there's also green tree python data that we've been working on, and also in talking to other people, things like that. I have some other things that I'm chasing, and I can say that what I'm seeing is the most exciting stuff I've ever seen for green trees. I think that this is going to work, but those of you that have been waiting like a year and a half for me to get this working again, you're probably just rolling your eyes like
right.
Before. But I will know.
Primers in today, right? So we can start trying.
I just got primers in today. Yeah.
Yeah. I know, because there was somebody who asked me at Arlington, and at that point, I think y'all thought that the last batch would be the one. And so I was like, well, just, watch the next episode. I can't say when, but, maybe. So thank you for holding on with us, green tree keepers like y'all have been.
We're feeling good enough about this. Ben, correct me if I'm wrong, but I don't think that I am. That if we do hit it, it's probably going to open doors onto multiple other python species as well. So in very quick order, we could, like dominoes, start picking up carpet pythons and blood pythons and all kinds of other stuff right behind it.
Ticks and bloods and burns. Mm-hmm. People have asked for pretty much all of the python species.
Yeah. This is actually regarding the last topic with ultramel, but it was an important question from someone. From HB Reptiles, do ultramel tests that failed for A need to be sent back in to test for B?
What you could do, we've kept those. So if you got a result that said negative for A and we were not able to test for B at that time, just let us know that shed ID and order number, and we will look it up, and we should still have the shed here, and we will run it for you and get you that result.
Good stuff. Thank you. All right. So back to chondros.
So along the same lines, I thought that I had this working, back in like last October. And then when I did a bigger run, that's why I started not offering something until I run at least 100. When I did a bigger run, it wouldn't work and I couldn't get anything to work well. And we had to cancel. We had to stop selling the green tree python sex determination test. Mm-hmm. But for those of you that did pay for a green tree sex determination test a year ago, and we had to tell you, sorry, we can't do it and refund your money, we did also save all of those sheds, and we will be running those and giving you your results. And since you had your hopes raised and then dashed, and that's our bad, there won't be any charge for that. We've already refunded you. We'll run it, give you your results, and there's no charge because that's our bad.
All right. We do have a couple miscellaneous ball python morph related questions if you wanted to hit those before we go, or to the next subject, I mean.
Sure.
Yeah. Let's see. Well, now I lost track of them. This is why we star them in chat. Let's see. Just from CG asking for a clarification. A little late on the Blue-Eyed Lucy complex announcement. So Phantom and Mystic did end up being the same gene?
So far, yes.
Okay.
Dr. Seidel has done a fair amount of testing. I know that she's not to the point she's ready to publish yet. But so far, what she's seen is Phantom and Mystic. She's not able to tell genetically that there's a difference at this point.
Okay, fair enough. And then from Dominic Taylor, any luck making progress in the banana or coral glow, determining if it's a male maker or a female maker?
So that's a test that we definitely could design. My fear is that it would be expensive enough that it probably would not be worthwhile for people. We'll see. So because banana and the sex determining gene are far enough apart that 6% of the time there's crossing over in between them, we would have to have some kind of, it be called like a haplotype kind of test. So it's a much more complicated test than all the other tests we're doing. But we'll see. We might be able to figure out a way to do it cheap enough that it would be something that would sell. But we also may be able to, anyway, we'll have to think about that. It's definitely doable, but might not be cost effective.
Fair enough. All right. So do we have anything else before we start moving on to what our goals are for 2024? Because I know we wanted to talk about that.
Someone was asking, let's see, Geeked was asking, have we looked into genetics of red and yellow neonates in green trees? That's something that I've heard people talk about, but I honestly don't know. I think that that red is recessive and yellow is dominant, but if someone's a green tree python in here and knows, feel free to jump in. Or if it's something they don't know for sure yet, let us know. But I think that that's kind of from enough breeding. If you breed a red to a red, I think you get all reds. But if you breed yellows together, that's not necessarily the case, if I remember right.
Okay. Cool. So let's see. Now we were wanting to go on to this subject, right?
Yes. Oh, actually, before we go off of sex determination, so green trees is one, and then pythons in general. But like we talked about many times, blue-tongued skinks, heloderma, and varanids, those are on our list too. I think pythons will be our main focus for 2024. But if we end up flying through those, like I hope that we can, it'll just depend on how lucky we get. It was a big difference between ball pythons and green trees, but I'm hoping once we have ball pythons and green trees figured out that the others will be more simple. But anyway, we'll see.
Sex determination tests, right?
For sex determination, yeah. So if we can get through the pythons, then blue tongues and heloderma and varanids are ones we are interested in as well.
Cool.
All right. So yeah, then I think, Sean, maybe you want to jump in on this. We were talking about as things kind of slow down some and we start to kind of shift gears, we have more time to focus on developing new things. Obviously, what we just talked about, the morph tests and the sex determination tests are at the top of our list, but we've also kind of talked about some ideas of how to start something new and exciting for 2024, where we allow you as keepers and breeders to be a part of the development and be able to have your name associated with genetic tests that are developed. And I guess we're going to call it like a sponsorship, be able to sponsor a new morph test or doing whole genome sequencing in depth to be able to start working with a new species like hognoses or boas or, some gecko species people have reached out for. So those are some things that we're kicking around. Is there anything you want to say as an introduction, Sean, about that?
Okay, deep breath. Here we go. I'm always the guy who's got to be the bearer of bad news. Okay, so I mentioned in a live stream about 6 months ago that we were going to get to a point with ball pythons where we need to put the brakes on a little bit. We're getting there. We're going to be pretty close to probably 50 mutations by the end of the year, if not already there by the end of the year or right after the end of the year. That's kind of always been our goal for the first year. I think we're going to either hit it or be really damn close. And then we're going to slow down a little bit. We have so many other people asking us to do so many other things. The thing is all of these tests that folks want done cost money, right? They cost a lot of money depending on what they are. So let's stick with ball pythons to start with. We're actually going to start a program where, look, we're going to keep working on ball python mutations. We're not just going to stop, but we're going to stop making it our primary focus for a while and try and get some research in for some other species as well. That said, if there's a mutation that a particular group of breeders or just a group of hobbyists wants to pitch in on to help us get it past the finish line, we're going to have an opportunity if you guys help us sponsor. And really in sponsoring, all that is you'd be prepaying for tests at a discounted rate, particularly if you're sending us sheds to help us get there. And there's lots of things we can do in exchange for that. You'll have your name and your logo associated with our company and on our website as helping to be one of the people who helped found that mutation test. As I said, you'd be prepaying and among the first people besides Ben and I to have your sheds run and tested before anybody else because you helped us get there. We're trying to hit a target of around $5,000 for every new mutation. Nobody needs to donate $5,000 by themselves. You can offer up to 20 paid tests per person, and that'll be at essentially $50 a test, which is $15 minus sales tax. So probably more like $20 less than we sell for right now. The benefit there would be obviously you get results before anybody else on your animals. You would be associated with that mutation forever. You would literally be able to say, I'm one of the guys that helped get that gene on the map. Anybody who's donated a significant number of sheds, we appreciate that too. We're going to give you guys some credit as well. But then all of these eventually will also be in a scientific paper. Anybody who helped us get to some of these new genes is going to be credited in those papers as well, in the acknowledgement. So I'm always hesitant to ask for money ahead of time for things. But I think like with the green tree python test, as we've proven, if we try to make something happen and we fail miserably, I don't mind giving people their money back. However, one of the things that we want to focus on is other species and other mutations within other species this coming year. And we would just like some feedback, maybe financial feedback from the hobby that says, hey, this one's really important to us. And these 5 breeders have all pitched in $500 or $1,000 or whatever. We don't have to get to the $5,000 number, but if I've got $2,500 or $3,000 across 3 or 4 different breeders, like, no, we want lace and we want it right now. Guess which one we're going to work on first? That's going to be an indicator that we've got to try and tackle those first. There's lots of other stuff that we probably could have already had tests developed for. They're just things that aren't going to be super popular, pastel and stuff like that, you know, they're going to help us hit that 50 number by the end of the year. There's some stuff that's not as popular, like lace and things like that. And just because it's a little bit more rare, if you want us to focus on those, we need sheds. And if you guys can throw some money in it, it would be helpful. And we're giving you all of the benefits that are listed on the screen.
Yes. Yeah. As a thank you, we want you to be able to prepay. You'll get something back. It's a lot like Kickstarter. Any of you that have checked out Kickstarter, you're paying for a service, either a good or a service. In this case, a service. You're prepaying for it before it's ready, and then as soon as it's ready, you get served first, and you're getting at a discounted price. And then above and beyond that, we will set up a web page for each morph. So we'll have a monsoon. We'll have a monsoon web page where we'll say, who sent in sheds to us, their names and logos. If you sent a significant number of sheds in for monsoon, we'll have it for ones we've already done. Puzzle, several people sent in lots of sheds for Puzzle to get us there, and Monarch. So we'll do that as a thank you for people for sending sheds in. But then this sponsorship would just be another thing that will be on each of those morph web pages. And that's another chance for you to be able to have your name associated with that morph. So it's the morph you work with. You've got a few animals, send some sheds in to help get it developed, but then you can also prepay for your paw sets that are coming, and you would be among the first to get those run.
It's just like anything else. We get requests literally every day for all kinds of stuff. And ball python mutations are at the top of the list of those requests. But again, like anything else, the people that are actually willing to pitch in to help you get there are pretty few and far between. So we're asking those of you that really want to make this work and make these happen. If you could step up, you're basically paying for tests ahead of time, but that gives us an actual direction to go instead of just going by word of mouth. We want to start working with the guys that are really trying to help these projects along. So anyway, that's what this program is for.
All right.
And then one other thing that this program is really good for, and I've had 2 or 3 people, actually one at Tinley we talked to directly. If you have a new mutation that's only in your collection, or maybe you and 2 or 3 other people are working together and it's a new mutation, that's another chance where, between the 2 or 3 of you, if you can get $5,000 together and enough sheds. So you had to have like 2 or 3, maybe 4 generations of breeding to be able to have enough sheds. We need, 30 or 40 that are either heterozygous or homozygous for that new mutation. So it can't be like a brand new thing that popped up and you have, 1 or 2 or 3 animals. You have to have bred it for a few years. But once you're to that point, if you have enough interest to be able to put the money up, then, you all, if it's just you or 2 or 3 people, it's a new mutation, your names would be on there as the people that donated the sheds and the people that sponsored that program. And that would always be on the website. And like Sean brought up, when we do publish things in the future, you'd be in acknowledgments and scientific papers as well.
Yeah, that's pretty cool.
That's certainly a good way to get the word out on a new mutation. If you say, hey, I've got this new mutation. Oh, and by the way, RGI has a morph test for it. That's pretty legit.
Yeah.
It's not something, like I said, you can't just have an animal pop out and, oh, here's a test. We can't develop a test on 2 or 3 animals. We have to have several. So everyone would know if you're able to have a test developed, then that means you've worked on that morph for a few generations, and you've done the legwork by breeding and by doing the DNA sequencing and testing. So it's a pretty cool way to do it.
All right. So was there anything else on the sponsorship program that idea, like when you said this would probably be a 2024 thing that we'd start making posts about it, right?
Yeah, probably. If there's something, if you start talking in one of the, forums or groups or whatever, and, people are excited about doing this, you already have 4 or 5 people that would want to jump on, but feel free to reach out to us. But this will be kind of a, by invitation thing. What I'll do, like for Monsoon, for example, if we decide, I'm not saying we for sure are, but if we decide to do it for Monsoon, I'm going to look at my list of people that donated Monsoon sheds, and I'll just start with that list, the people that sent in the most Monsoon sheds, and I'll reach out to them and say, hey, we're going to make Monsoon a sponsored test and give them the first chance to be able to prepay for some tests if they want to, and be a sponsor for Monsoon. That's what we would kind of do if it's something that we drive forward. But certainly if there's, a group of you out there that want to do this for, someone was mentioning blackhead, any of the other ball python morphs, you've got a group, you want to send that in, let us know. And like Sean said, if there's people excited about it, they're sending us sheds and they're willing to prepay for testing up front, that's going to get done faster, because that's what we really need. So far, we've been paying the bill for everything, this development costs up front. So having this prepay helps us to pay for the development, and then in the end, you still get tests at a discounted rate. So it's a win-win-win in my mind.
Well, I just want to say one more time. I've said it once already, but we're looking at having somewhere between 40 and 50, maybe even over 50 tests here by the end of the year. We started a year ago in October with 6. So we've been busting our butts, right? So the reason we're slowing down, and I've also mentioned this in the past, is, we've been pitching a product called the panel, which is all of our tests. Well, we warned you guys about 6 months ago that somewhere between 20 and 30 tests that the panels would not run as cleanly. We're there. Part of the reason the panels are taking longer to get back results-wise to you guys is that a lot of them are not running well the first time. When I say not running well, we get really good clean results on half or 2/3 of the genes, but then we have to rerun on the next run the remaining 3 or 4 genes on that panel. The more mutations we add to the panel, the more complicated that gets. Now we have a solution for that, and that solution is going to launch January 1, called the DNA registry, which we've talked about in the past. However, I just did want to point out that I say we're slowing down. It doesn't mean we're stopping. But once we get to that 50 number, we have to find another way to run the panels because it's too many to do it the way we're doing it now, too many genes rather. And then going forward, hell, I lost my train of thought. Going forward, that new panel test is going to take longer. So we're going to have to start breaking some of the more common kind of panels into smaller groups, which is, I think, the next thing we were going to talk about, right, Ben?
Yeah. So the first panels we'll have available when we do break them out, which could be January 1st, somewhere around that time. We'll certainly announce on here and in our social media when we do so. But yeah, it would be something like yellow belly complex, albino complex. That would include albino, ultramel, lavender albino, monarch, any of those albino-like genes. Then blue-eyed Lucy would be another complex. And then in the future, it could be like the 8-ball complex, the black-eyed Lucy complex. So as we get more of those tests figured out, if they're groups that, people would maybe want, this looks like it's something in the blue-eyed Lucy complex, I want to know which one it is, or it is a blue-eyed Lucy, I want to know what, of the blue-eyed Lucy mutations makes this up, then you could just run that small panel. And so it would be a lower price, not as much as the panel right now. But, it'd be a simple way to be able to do that. What's that?
I said more of the key genes that you're looking for rather than hitting all 25 or 30. You're just hitting the 5 or so that are in that group. Actually, our next slide was coming up. It was about the species sponsorships, right? And there's a question actually about that on the board right now from Amanda Hempel.
Yeah. Mm-hmm. Yes, the sponsorships are a great idea. What are the genes that you're wanting to look at first for hog noses? That was one, right? Yeah.
Yes. So the way that would work, we've up to this point mostly been talking about the new morph tests. And so for us to develop a new morph test, we need to have a in-depth whole genome sequence for the species we're going to develop the new morphs. And for Western hognoses, we don't have that yet. And so for like Western hognoses or boas or chameleons or geckos, any of those that we don't have a really good in-depth whole genome sequence for yet, that would be the first step. And Sean knows better than me the actual numbers, but our kind of round number for doing that for the ball python, that ended up being around $50,000. So it's a pretty large investment. And so we did that. We had many people help out with the GoFundMe. We set that as a goal to be, I think, $15,000, but ended up being much more expensive than what we were thinking. And the GoFundMe was significantly helpful and made it faster. But yeah, it's a significant amount of money. So for a new species like hognoses, the first thing that we would want to do is we would pre-sell tests to people for $100. So this would be a little higher since we have to cover the whole genome sequencing. But you could buy prepaid tests for $100, and then those would be, we would have you all kind of vote. So once we reach, let's say, and we're not expecting the prepaid sales to get all the way to 50,000. If it did, that would be awesome because everything covered ahead of time, and then you all will still get tests for what you paid for. Once again, a win-win. But even if we got halfway there and we as RGI need to pay the other half, I think Sean would make that decision, but I think that halfway there, we would jump on that and we'd pay the other half and make it happen. So for that, once we decided, hey, we've got enough to go, we would just reach out to the people that are the sponsors for, if it's hognoses, we reach out to our hognose sponsors and say, hey, we've got enough money and we're ready to go. What are the 3 morphs that you would like us to design tests for first? And so that's kind of would be in y'all's hands, whoever are the sponsors. And then we would design tests for those 3. And then as soon as they're ready, those of you that are sponsors would get your sheds run first.
Cool.
So again, like I pointed out with the mutation sponsorship, this is just a bigger version, but it's exactly the same thing. To get into a new species and just to start developing tests the right way, it's really expensive, really, really expensive. $50,000 is not an amount that is where we're going to make money. It's an amount that what it's going to cost us to get the genomes done, and then to take as many hundreds of sheds as you guys can send us that are representative of different mutations, and send those in for sequencing too. Though every one of those sheds after the full genome is run has a significant cost attached to it as well. So it's expensive. And then once we get that data back, then we can start analyzing it and designing tests, which is again more cost. So we put a $50,000 number on there because that's honestly about what it would probably cost us to break into any new species. And two things as a business manager important to me. What do I think will sell? Hognose are very popular. And who do I think will step up and do that first? Well, when I was in Daytona, there were an awful lot of very large hognose breeders there, and they all pulled me aside and said, we want this desperately. How much is it going to cost, and can we pool our money? Well, now's the time, guys, okay? Going into 2024, we're putting the brakes on ball pythons a little bit and trying to work on other stuff. So if you guys want to start talking amongst yourselves and figuring out ways to help us raise some money and maybe help us get our hands on some wild type sheds, we can get a genome started. And we're talking about months of work, right? This is not going to be you guys plunk down some money and 3 months later we have a bunch of tests. You're probably looking at a minimum of 6 months before we get any kind of like real progress. But once we have that genome, we have all the puzzle pieces that we need to start actually working on this. So other people have stepped up and said they want to do it in other species, but one of the flip sides of the coin is even if they put the money up, I don't know if it would be worth it for us right now because we're not going to sell a bunch of tests, right? Hognose is a different story. I've gotten so many requests about hognose snakes that I would really, really, really like to make that our next species. But again, it's folks calling us and donating a little money here and there that's going to help lead us down the path of what the hobby wants next, right? So that's it.
And one thing I saw Randy from Dark Cloud Reptiles mentioned earlier, it's been a while, but he said donate an animal and auction it and get use that as money.
That works.
Yeah, he literally did that at the Pomona show and raised, he donated a $5,000 animal, raised a nice chunk of money, and I went directly 100% into our development costs. Any way you can do that, that's awesome. And like I said, we want to figure out a way to make sure that we thank you on the backside. So we'll offer you, prepaid tests and have your name associated with that new genome and/or new morph tests, whatever it is that you're helping with. We want to make sure that there's a legacy there.
Absolutely.
Hey, I just want to answer a question that I've seen pop up a couple of times because I'm not sure how people are meaning the question. People have asked about DG a couple of times. Yeah. And I don't know if they're asking about the DG project that we had everyone participate in starting in June and that ended a few weeks ago, or if they're talking about, is there any new information in general on DG? Now, if you're asking about the research project, yes, that's done. We've received all the sheds that we're going to receive, barring one or two that I gave a little permission to send them in a little late. We are over 525 sheds now.
Wow.
Donated sheds. So we are about to learn a whole lot more about Desert Ghost. And results for that will start going out over the next month to 6 weeks. We're going to make that spreadsheet public so all of you can see it. It's all 520 some odd lines. The only thing I'm going to take out of the spreadsheet is everybody's email address because that's kind of personal information. Yeah. But the rest of it is going to be who produced it. Here's a photo of the animal. These are the results, head A, homozygous B, whatever. And all of you super sleuths, if you want to get in and play around with that spreadsheet, you can download a copy of it. And maybe you guys will see something that we don't see and will give us a direction to chase down another rabbit hole with DG and see what else we can learn. But that's the whole reason we did this project. I wanted as many diverse sheds. We got sheds from all over the world. So awesome. How are we doing on that? It's closed. All of the spreadsheet has been built, save again, maybe another 12 to 20 sheds that I'm still expecting in. Half of them have already been prepped and are in plates and just waiting to be run. So, and Ben will be the one to actually prep and run those. I think the first plate's probably going to get done, in the next 2 to 4 weeks?
Yeah, should be.
Probably another plate every run, and there's 96 samples in each plate. So over the next few months, you guys will start seeing results updated on a public link for all of you to see. And that's how that's going. And I hope that answers most of y'all's questions.
Yeah, and as far as if you were asking literally the next paid results, we talked about that at the very beginning of the episode, but those would be starting to come out on Tuesday, and through most of next week, those results are coming out. But I think, Sean, you answer. I would imagine most of them are thinking about the free testing.
Come up 4 or 5 times, and I just wanted to address it so people don't get frustrated. We're on it. The hard part's done. The hard part was receiving all the sheds and adding them to the spreadsheet. That took forever because we have to upload the picture to a site and then link the picture with a thumbnail to the spreadsheet, and we had to do that 500 and sometimes. It took a minute, but now we're done. So now the easy part is stuffing them in strip tubes and sending them off to get them tested for sequencing. So we're almost.
The other big news for DG is we do, along with this batch of data that we've been talking about, had Puzzle and TSK and Monarch and Green Tree Pythons. We also had another big group, like 40 or so Desert Ghosts and Het Desert Ghosts on there. So I'm hoping within the next week or two, I will have another thing to chase and be able to improve our DG test in the short term as well. And that's another reason. So first of all, we've been waiting for all the sheds to get in, and then for poor Allie to make it through all 500 of them, listing them all and getting the pictures in the right order and then prepping the sheds. It was a massive amount of work. I'm sure Sean,
the busiest time of the year. We were getting crushed with paid samples at the same time. So
we have this question about DGC. So that's what I'm hoping is in this batch of data it's analyzing, it's churning. We have to upload these billions and billions of base pairs and let the bioinformatics software chew on it, and it takes several days. So it's chewing, it's churning right now. So I hope that I will have something to chase here in the next week or two. Something's interesting.
I want to touch on something that you just said, because I don't think people realize that when we pull this data down and we put it through the software, Ben's not kidding. It takes 2 or 3 days for the computer to process all of the billions and billions and billions and pieces of information. So it amazed me when I learned about it. I mean, I've never seen anything take more than a couple of hours on a computer, much less days and days and days to return some results. So it's anyway, I just wanted to touch on that because you gloss over it because you're used to talking about it all the time because what you've done for a living for the last decade and a half. But for most normal people like me to understand that a program has to run and process and just crunch numbers for 2 or 3 days tells you.
These are powerful computers too. Right. This isn't like a little computer, a little laptop. This is a powerful computer, and it takes it several days. We had one analysis, it was on another species. It wasn't on ball pythons, but it was just a simple parentage testing for some turtles, and that took almost 4 weeks. It was training on that. Yeah.
Wow.
Yep. It was just a ton of information for a bunch of individuals, and it just took forever. So, and like I said, that was actually multiple multiple computers churning on that for 3.5 weeks, almost 4 weeks.
That's incredible.
And one way I like to show it to people is put up, a bunch of sequence and then start scrolling down, and you see all of these base pairs flying by, but the little, thing on the sidebar, it's like not moving.
Oh no.
So when you're talking about 1.5, so snakes, that's 1.5 billion base pairs, 1.5 billion A's, T's, C's, and G's. And, even if you're just looking at one chromosome, that's a couple, 100 or 200 million base pairs. Just to scroll through that, just to do a control find and look for, 20 base pairs that you think are unique in somewhere on that chromosome, your computer's gonna have to think a little bit to be able to find it in just one chromosome, let alone a whole genome. So it is pretty mind-boggling. It's a ton of information, and we're very, very lucky that people have worked on the bioinformatics, the computer side, to be able to handle those massive amounts of data. Now it's easier to produce sequence data than it is to analyze it, because we can produce such massive amounts of sequencing data, but then to be able to make sense of it when it's so massive, it's a different beast.
I didn't mean to sidetrack it too much, but you tend to brush over these things, and I just wanted to make sure everyone was aware that it's a massive amount of data anytime we're working on something new. Speaking of bioinformatics, Patrick McKnight just jumped in and said hey to everybody. So, hey, Patrick.
Hi, Patrick. We had a picture of you at the beginning of the stream. You and Ben sitting there at Tinley on your laptops.
Yep. That's another thing on the list back here to talk about. We're working on right now, and Patrick helped me start down that road to be able to make the analysis faster. So that's another thing we're improving, and we'll do that on multiple fronts. Another really big part to that is we are working to automate it so that when the calls are made in the software for tests, that automatically gets sent to our vendors, to MorphMarket and Clutch.
Sorry.
Two pretty boys learning how to use computers. I don't know which part of that is less true, learning how to use or being pretty boys.
I'm seeing uglier, you know.
Things are getting weird. Stay on point. Stay on point.
Okay.
What's next?
Question about boas. Do you know if you'll have any results on sharp or call albino boas?
So we would love to work on that. I'm still gathering sheds for that. So I really would need to have, since it's likely that most, if not all people don't know for sure if they have one or the other, maybe have, het for one and the other, it could be a mess. So we definitely need to have at least like 50, if not like 75 or 100 sheds from albinos, or for sure 100% het albinos in boas to really start that. And we don't necessarily have to have a whole genome for the boa to start on that project. So that, big chunk of money, tens of thousands of dollars up front, we don't necessarily have to do that because albino is pretty well established across mammals and reptiles. We know what genes to look in, but we need the sheds. So if you're interested in call and sharp albino boa tests, talk to people that have them and send sheds into us. And once we get over 50, then we'll start getting serious. And if we get to 75 or 100, then we'll definitely do that.
Yeah. Before we move on to more questions from the comment section, was there anything on the whiteboard behind you that we missed?
Discounts.
Let's see.
Discounts? Oh, hey, we like discounts.
My favorite topic.
Yeah.
Discounts, sales.
So I promised everybody months ago that we would do this starting November 1 definitely through MorphMarket and probably through Clutch as well. Although again, as I mentioned, they are going through a big software update. So when they're done kind of recoding some of that, they already have our stuff built in, but they're probably not going to want to turn it on until they make sure their software is up and running and good to go. That said, starting November 1, anything 10 to 99 sheds, you now get 5% off on anytime. Anything 100, I shouldn't say that. It's not sheds, it's tests, right? The discounts are going to be based off the number of tests. So you can send me, 5 animals with 2 gene tests each, and that qualifies for a discount. If you order 100 tests, and we should even really call it a test, we should call it an item, right? Because a panel would be 1 item, not 30 tests. You'll get 10% off. Those are going to start automatically and will be on all the time, 24/7, all year long. And coming into slow season, which is why we're trying to launch this in November, we're going to be adding another 5 and 10%, maybe sometimes even 15 or 20, depending on how slow of a month we're expecting it to be, to try and drive revenue and have folks send us larger groups of sheds so that we can keep our plates full and keep sending sheds on a regular enough basis that we don't have to slow down on the volume of runs. So anywhere from 5 to 25% off based on the time of year, but you're looking at 5 and 10% all year long, starting November 1, from now until essentially as long as, Ben and I are in charge until the end of time. So we promised it. We know it's not a huge discount, but we did that on purpose. We would like to drive the bigger discounts for the slower times of the year. January, February, March, I'll be honest with you guys, nearly killed us last year. It was bad. We dumped a whole lot of money into a whole lot of research and did not get a lot of sales in those months. And we want to kind of train everybody, for lack of a better term, especially some of the bigger breeders. Look, don't wait until 3 or 4 weeks before a show to send me a bunch of sheds. Do it in January, February, March when things are cheaper. And It helps keep us busy and helps keep our lights on and helps us feed the kids. And in exchange, you'll be able to save a significant amount of money. Additionally, one of the things, and I would like your comments down below or in the comment section, however this works on a live stream. I'm seriously debating through both of our vendors. As a business owner in Texas, Texas doesn't mess around with sales tax. They don't really care what the laws are in other states. And let's just say they're not very nice. And my other companies have been audited a couple of times over the years. It's a very uncomfortable process. So we've charged sales tax on everything through both of our vendors. And we know that makes a lot of you unhappy. So we're going to kind of compromise with you guys. The prices on our tests are probably going to go up just a little, but there's no longer going to be sales tax. Basically what I'm telling you is I'm going to roll the sales tax into the price of the purchase of whatever you're purchasing, and it'll be less than you would have paid had you bought the test and then paid sales tax. So we're going to discount the price a little bit, but then add a couple of additional dollars to cover sales taxes. That way y'all don't have to mess with it anymore. It becomes an accounting issue for us. And yeah, so there's that. And then you can take advantage of the quantity discounts on top of it. So it just makes sense for everybody. So I think we're going to just
throw it in there really quick. And you know, like Sean was saying, run your tests during the slow months, but also if you know you're going to be producing over 100 that you want to have tested, or you know you're going to be producing at least 20 that you want to have tested, if you pay for those in January, February, March, you don't necessarily have to run them then, but you could pay for them when we have that discount. So if you watch when we have the deepest discounts, the 20, 25% off, and then pay for what you know you're going to for sure use anyway, then you've essentially prepaid, and then as soon as those animals shed, you just send them off, but you get it at a discount. And then like Sean said, that helps us during the slower months that we have some money coming in, and we'll thank you for helping us to keep all our bills paid during the slower months by giving you that discount.
Well, and we have to fill plates to send them off for sequencing. So, we want you to send them in when you can, but obviously if you're not going to have babies until spring, it surprised me actually to the point that I brought it up to both vendors, and it sort of shocked them too. A lot of people are ordering these tests 2, 3, 4 months in advance and then just sitting on them, whether they pay for it in advance or not. I know in MorphMarket, you can go in, create a bunch of animals with potential tests, and then either pay for it at the time and wait and take advantage of the discount, or you can just create the orders and then 3 months later, when you do actually get offspring, pay for it at that time. But it would be more expensive. So there's multiple ways you can do it, but it surprises me how many people plan that far ahead. I mean, we get stuff 4, 5, 6 months out. I mean, I was still up until a couple months ago getting stuff from last October.
Wow.
Where people created orders in that very first push and didn't actually get a shed till almost a year later. So, and they don't expire, by the way, guys. If you purchase a test, it's good until we get a shed in and we close the order out. So
yeah. Cool. I guess that's related to Pat McKnight's question here. You have to buy them all at once, and will you have an option to pre-purchase? So,
yes, yeah, definitely can pre-purchase. Yeah, if you know you're gonna, like I said, if you know you're gonna run over 100, then you can go ahead and pay for 100. And if you towards the end of the year, you're gonna have like another 20 or something, reach out to us, and we'll make sure we can continue that discount for you. As a thank you. If you're running lots of tests, we want to make sure we can get you a quantity discount. And then if you can pay for it during the slow months, we will thank you by giving you that deeper discount.
Awesome.
So in short, 5 to 10% based on quantity forever, and additional discounts during slow season because we need to stay busy for multiple reasons.
Yeah.
Okay, I'm done with that part.
Awesome. You starred Not Your Fault, Texas Gonna Texas.
Yeah. Okay. So I don't know if you guys are in Texas, but yeah, Texas doesn't mess around. I've been a small business owner in Texas for almost 30 years now, and I do know the one thing that they don't care about other states' rules. If you sell something out of state, they expect you to pay sales tax on it, period. Wow. So I know there's some gray areas in the law, but I've never found an attorney here that was willing to fight the state over the gray areas. They're just like, dude, it'll be cheaper.
Yeah, fair enough. All right. So we have some other questions. Let's see, from Pip Stalkers, is work being done in the spot nose and spider complex? Sorry if you already talked about it. They're at work and just hopped on. I don't think we talked about that. So
we haven't talked about that one specifically, no. So yeah, we definitely are working on that some, and I've talked with both Hannah at Eastern Michigan and Heather in Canada. I think one or the other, or both of them, are interested in working on that as well. So it's in the works. So the black-eyed Lucy complex, the 8-ball complex, the spider complex, all those that at least how they breed look like they are allelic to us. We will be able to figure out if they are actually on the same gene or nearby genes, and what exactly is going on. That's all work we're very excited to do, and I have made some progress on the spider complex. And the black-eyed Lucy complex as well. Fire, I have some good information on, but not all the way there yet on any of them. But it's coming.
Cool. Let's see. We did just hit that 1 hour mark in our live stream, so we have 4 more starred comments left. Do y'all want to hit those and maybe call it a wrap, or do you want to keep going?
I see one I want to address real quick.
Yeah.
Christine, or is it Kristen or Christine? I don't know.
Kristen Dion, I think.
Yep. So the short answer to that question is no. The vendors don't want us to do a create your own 5-gene panel because programming wise, it's a little more complicated. However, if you look at how we do genes now, you buy a single gene test for $65. Second gene's only $25. If it gets to the point where people are really wanting to do 3, 4, 5 genes, we could probably build something that would be first genes, $65 or whatever the price is, and then they cascade down from 25 to 20 to 15 or something like that. I mean, a full panel is $120. The 5-gene panels are probably going to be somewhere around $95. I mean, if you can get 3 or 4 genes for that just by selecting them separately rather than creating your own panel, that's probably going to be the way to get there. So the answer to your question is not officially, but unofficially, yeah, you can probably do that, and you're going to come out to about the same price a 5-gene panel would have cost anyway. So,
all right. Let's see. From Heathen Hatchery, Brian, is there going to be a Mandarin test, or where are we at as far as like shed submissions? There.
Need more sheds. Yeah.
More sheds.
At shows, I've had 2 or 3 people tell me, and I saw in the comments earlier people bringing up paint also. Both paint and Mandarin, people have said they'll send sheds, and I have gotten a few, but we need more for both Mandarin and for paint. We need more sheds.
You got it. Yeah, I think I saw something about paint in there too. So yeah. Likewise, going back to the spider complex, are there any sheds that are in that complex that are higher on the wish list that might help out? Some
of the less common ones. I think Cypress is part of that. That's one I don't have a heck of a lot of sheds for. Okay. I've got plenty of spider sheds for sure. The really common ones. Even blackheads, I should be able to find enough blackheads, but if you have super blackheads, those would be worth sending in. That helps us to have the homozygous version of any of them we're developing a test for. That helps us get there faster.
Yeah.
But yeah, the less common ones in the spider complex would be helpful.
All right. And this one is a more general question about shed tests from Sarah Gilmore. I use cocoa fiber and it gets stuck on the shed. I shake off a lot of it and brush some off with a makeup brush. That's really detailed. But I can still see some specks of dirt. Is that still going to be usable?
So the thing that helps the most with this is if the majority of the shed is good, like they don't shed in the water bowl or crap in it and accordion it all up on top of that crap or something like that. Yeah. All of us know happens sometimes. If you have a decent amount of shed that's flat and clean, even if there's some shavings or different things in there, if you just find the piece that's the cleanest, best, size of a quarter or inch square, however you want to think about it, just tear out that little small piece that's the cleanest and just do the best you can, and definitely appreciate that if you do take the time to. You could also run it underwater and just with your finger pull that off and then just set it to dry. It's okay for it to be in water, it just can't be in it very long. But if you get that cleanest piece and there's a little bit of funk there of bedding or anything, you could run that underwater, rinse it, get it with your fingers, and then just set it out in a dry place and let it completely dry for a day or two before you put it in a Ziploc or an envelope or anything. I can tell you that I have noticed sometimes the few that do fail in a run are ones that have chunks of junk in them. So it is worth spending a little time. And then on our side, when we're taking the really small piece, sometimes we're taking like a single scale or 2 scales if it's, a larger animal. So we're going to do our best to avoid those chunks as well. But yeah, less of that extra stuff, the better for sure. And if it's a funky color, if it's, kind of a fluorescent yellow or something, don't send that. It's not going to work. No. Wait for the next shed or look for a piece of the shed that's not like that. And if you send a whole shed in because you want to make sure we have everything we need, but part of that shed's really crappy and it sits in a Ziploc bag for 3 days or 4 days while it's shipping, then it's, kind of messing up the rest of the shed. So I really, really like to see just that quarter size, inch square, however you want to think about it. Just tear that small piece out, get it as clean, flat, and dry as you can, and send that. And that's going to give you your best chance of getting results on the first try.
Exactly. Like just make it as pretty as you can, sharing a relevant meme here, just because it is, it works. That's my rat snake Clover. Okay. And then, oh yeah. Jody Lopez says, Sean's looking like a Star Wars fan. Yes.
I was wondering if anyone would notice the Boba Fett hat.
I actually for many, many years was a member of a group called the 500 First. It's a professional Star Wars costuming group. I was Boba Fett. That helmet, cool. Along with my whole costume, I PVC vacuum formed myself 20 plus years ago. And that helmet in particular has the signature of every person that's ever played Boba Fett up to current dates. So whether cool. Boba Fett is a kid or Boba Fett is an adult. So yeah, I'm a giant Star Wars nerd.
Nice.
Sorry. Yeah.
Cool. All
right, I have Boba Fett tattoos all over my torso, but I'm not taking my shirt off for you.
Oh.
You got to donate. You got to be like a super sponsor for
super sponsor. Exactly. I'll take my shirt off on the camera. That's right. Shave my hairy chest so you can actually see the artwork.
That's dedication. Yeah.
Yeah. We did have a couple of quick things that we wanted to talk about too.
Yeah.
So we are trying to make our software a little bit faster on our end, but we're also working on auto updating results into MorphMarket and into Clutch. That's 2024 stuff. That's to help us prevent human errors. It's a big deal. If you consider there are 30 genes that we're testing for, and I have 100 panels, that's 3,000 genes. And I personally do most of these, that I have to manually update 3,000 individual genes across 100 orders. It's very easy to make a mistake, right? And so we're doing, I don't know, 500 or 600 tests at a time, right? And that's not the number of genes, that's just the number of samples at this point. So we're pretty busy.
Yeah.
In terms of tens of thousands of, yeah, of course, Patrick, derailed the conversation.
He's saying he doesn't want to update 3,000 things.
I don't think that. I think he's hard passing on me taking my shirt off. I may do it now just to piss him off. Okay. So we're really hopeful that we can get the vendors to help us absolutely flexing my nerdhood. Yeah, buddy. You don't see all of the really cool stuff over here. This is behind me where the dogs sleep. All my really neat stuff's off to my left.
Next live stream, Sean's basement.
Yeah, I'll pull the camera off and show you all my old school Dungeons and Dragons, World of Warcraft, Star Wars nerddom all over this room down here.
Yeah, let's go.
This is anyway. Software updates are coming. It should help with some of the errors on the back end. Should make it a lot faster. I mean, when Ben reports results to me, it takes me 3 days to get them all in the system. That's how many, and I mean 3 8-hour days of me sitting in front of a computer, clicking through, adding stuff. So when you guys are asking why it takes so long, every step of this is hours and hours and hours of work in preparing the sheds, getting them prepped for the run, analyzing the data when it comes back, translating the results, and then putting the results in the system. Every single step is multiple days of work. So 3 to 5 weeks is a pretty good turnaround time for us. I'd like to get that down to like 2 to 4 weeks, 2 to 3.5 weeks. And the only way we're going to do that is if we can start using software to upload that stuff. So again, that's another change for 2024. We're pushing really, really hard for that this year.
And that's really honestly another thing that helps these new morph and new species sponsorships help kind of to cover development costs for new tests, so that we can use the money we were spending on that, we can use that to help develop more automated ways to get all this stuff done quickly and more efficiently and more correctly. So there's not human error. So that's another good reason to move forward the way we're thinking and make things so we have more tests and faster results for y'all.
We're getting there.
Yep.
Well, I think my phone's getting ready to die, so I either got to plug in or we got to be done fairly soon.
Oh, okay. Well, wait.
Sean's showing off. There's one.
All right.
There's one of them.
That's cool.
Barbarian play on Boba Fett.
I love it. Look at the skulls.
That's a Frank Frazetta, not ripoff because he never drew this, but it's that style of artwork. It's supposed to be Boba Fett, but only in a barbarian style. And all my Boba Fett stuff is adaptations of Boba Fett over time, from primitive to postmodern, like beyond where you're used to seeing Boba Fett. So I'm like big time nerd, big stupid time nerd, big bad nerd. That's me.
There we go. We got some teasers from the Sean's tattoos and basement. All right. So before Ben's phone dies here, I'll plug. I'll plug Reptile Genetics Services. That's another way that you can help fund rare genetics if you're in the market for a ball python or some Honduran milk snakes, which you just had hatch out. I don't have pictures of them here in the slide, but a lot of those proceeds go toward it. And also, he just has some cool stuff in that collection. It's worth checking out. He's on MorphMarket. I think that's everything we got. So thank you so much for everybody who joined us for the live stream. I think we hit like 65, closing in on 70 people at the peak of it. So
cool.
Love seeing that. Good numbers. Like, comment, subscribe, share with your friends. If there's a clip you really liked or some shenanigans you thought were funny, feel free to, make a short out of it on YouTube or something. Poke fun at us,
Not taking my shirt off.
No shirtless Sean.
Now that I know you're pitching making shorts and putting it online, definitely not taking my shirt off.
Well, we can dream. All right. I guess we'll call that a wrap. Y'all got anything else before we go?
Nope. Just once again, thank you. This is definitely has surpassed what I was hoping, and I'm very excited for 2024 as well.
Yep. We're super grateful, guys. I can't thank you enough. This is literally a dream come true for us. We're glad you guys have seen value in it and continue to allow us to do this. So thanks again. We really appreciate it.
Seriously. All right. Well, good night, everybody, and we'll catch you next time. Bye.